interphase, there's 4 chromosomes that are single. Red and blue are homologous pairs, they share certain structural characteristics, including length and shape, and they carry the same genes in the same gene loci. And then in the S1 stage, they replicate, having a sister chromatid held together by a centromere, getting ready for meiosis. In prophase 1, spindles of the microtubules form, and the centrioles organize them, and the homologous pairs go on each microtubule. And then in metaphase, homologous pairs align on the equator, and the chromosomes become most condensed, and then crossover will occur between the red and blue, which exchanges genetic material between the non-sister chromatids. Afterwards, anaphase separates the homologous pairs and pulls them apart. And then in telophase, chromosomes are now in 2 sets at opposite ends of the cell. Then we go to meiosis 2, similar to mitosis, where centrioles organize the spindle microtubules, and pairs of sister chromatids attach to each of the spindle microtubules set. Once more in metaphase 2, sister chromatids align on the equatorial plate of the cell, and then separate again during anaphase. And then in telophase, as we see, a nuclear membrane forms around each tetrad, and each contains 2 chromosomes, so there's 4 daughter haploid cells. Non-disjunction occurs when chromosomes fail to separate at anaphase. As you can see, the diploid parent cell with 2 chromosome 21, when they have non-disjunction during meiosis, one gamete has 2 chromosome 21, and the other one has no chromosome 21, so it dies. And when it fuses with a normal haploid gamete, it turns out the zygote will have 3 chromosome 21, otherwise known as trisomy 21 or Down syndrome. The karyotype originates from a micrograph, a photo of stained chromosomes from a microscope. The arranging of chromosomes as pairs is based on size and structure of karyotyping, based on the karyotypes shown. Non-disjunction appears where there's 3 chromosomes on 21, otherwise resulting in Down syndrome. The last pair of long chromosomes indicate that it is a female. If there was one X and a short Y chromosome, then it would have been a male.
Meiosis begins by a pair of sister chromatids held together by a centromere. During prophase, chromatin begins to condense with supercoiling, and 4 chromatids from each pair of homologous chromosomes are called a tetrad, which is 4 chromatids, or bivalent, which is 2 chromosomes. During metaphase, homologous pairs are aligned to the equatorial plate. In anaphase, spindle microtubules contract, pulling the pair of sister chromatids, pulling the pair of homologous chromosomes apart. And then in telophase, each pole contains a pair of sister chromatids. In meiosis 2, it separates the sister chromatids. So in prophase 2, the sister chromatids attach to the spindle microtubule at the region of their centromere. In metaphase, the pair of sister chromatids are lined up at the equator of the cell once more, and at anaphase, once again, it pulls the sister chromatids apart. Finally, in telophase, the nuclear envelope material that remains within the cytoplasm reforms around the set of these chromosomes.
This is a homologous pair, and these are the sister chromatids. Molecules known as cohesins hold homologous pairs close together, and then afterwards, exchange of DNA begins. This is most notable in the chiasma, and in metaphase, the cohesins break down and the homologous pairs repel each other, particularly at the centromeres, but they still link together in the chiasma. And then in anaphase, the separation of the homologous pairs breaks the chiasma connections, and then the DNA exchange on the arms of the chromosomes is complete. And then in anaphase 2, the sister chromatids are separated into different cells. Some of the chromosomes are new recombinants of DNA containing part maternal and part paternal chromosomes. This effect creates new combinations of linked genes.
At metaphase 1, the purple and blue chromosomes will separate from their homologous pair of green and red chromosomes, respectively. However, there is also a chance the green and blue chromosomes could separate from the homologous pair of purple and red chromosomes. This is random orientation, meaning that all orientations are equally possible. The calculation of the number of possible gametes is 2 to the power of n. Homo sapiens have 2 to the power of 23. The topic assumes sexually reproducing populations as genetically diverse and that diversity within the population changes from one generation to the next.